Okay, this is the last part of chapter 3. This is a long chapter. So this is part 5, and our focus here is going to be on nucleic acids. Okay, so again, nucleic acids are going to be large, let me move the camera, large macromolecules, okay, organic, carbon-based macromolecules. They are polymers. They are made up of nucleotides. So here we go. They are polymers made up of these repeating units called nucleotides. What are they? So they are responsible for storing, transmitting, and expressing genetic information. They are going to determine what proteins are made in the cell. Okay, so very, very important. There are two types of nucleic acids. There is DNA, which is called deoxyribonucleic acid. For short, we're just going to call it DNA. And then there is RNA, which is ribonucleic acid. And again, for short, we're just going to call it RNA. So DNA is going to make up the hereditary material of the cell. When we talk about genes, we're talking about DNA. And these genes, this DNA, contains instructions for making proteins. And so proteins, again, are made according to the DNA, according to the gene. Now, RNA, what is that? It's the middleman. Okay, so RNA is going to be used in the process of making proteins. And so it's kind of the middleman. So we've got DNA, and then RNA, and then proteins. Okay, and I've got a slide to show that transmission of information. But before we go there, again, both DNA and RNA, they are nucleic acids, and they are made up of nucleotides. And so I want to make sure that you understand what the structure of a nucleotide is. These are the monomers, okay, that are going to be put together by dehydration synthesis to form the polymers that we call DNA and RNA. So there are three components to a nucleotide. There is going to be a phosphate group, which is shown here in yellow. Phosphate group is phosphorus attached to 4 oxygen atoms, and notice the negative charge on these oxygens. Okay, so it's going to be negatively charged overall, and going to be hydrophilic. Okay, and then we have a 5-carbon sugar, and I want to make sure that you understand this for this chapter, but also because we're going to spend a few weeks talking about DNA in really good detail. So the carbons are these points on the skeleton ring here, and then this carbon up here. So let me number them for you. This is going to be carbon 1. It's called carbon 1, or the 1' carbon we can refer to it as. Carbon 2, or 2'. Carbon 3, or the 3' carbon. Carbon 4, or 4', and carbon 5. Okay? And then the last component is the nitrogenous base. And we'll talk about the different types of nitrogenous bases. But just so that you realize that this particularly is adenine, which is one of them. Nitrogenous base, what does that name mean? It means nitrogen containing. Okay, so look at all the nitrogen. So all these N's here are nitrogens. Okay, so a phosphate, a sugar, and a base. Okay, this particular sugar shown here is deoxyribose. If there was another oxygen right here, this was an OH group instead of just a hydrogen, that would be ribose. So that's the only difference between those two sugars. And I'll show you that on one of the next slides. Okay, so again, a nucleotide, these are the monomeric subunits that form nucleic acids. And all nucleotides, whether we're talking about DNA or RNA, they're composed of 3 things: a phosphate, a sugar, and a base. Okay, so let's compare DNA and RNA. So DNA, again, is deoxyribose, deoxyribose, that's the sugar, and RNA is going to have the sugar ribose. So what's the difference? They're drawn right here. So again, we're going to look at the 2' carbon. So 1, 2' carbon. The 2' carbon here just has a hydrogen atom in deoxyribose. In ribose, the 2' carbon has a hydroxyl group. So that's the difference between the two. Okay. I should have actually said this on the previous slide. Let me back up just 1 second. Okay, let me move my head. I just want to show you where things are attached. So we always look at the sugar. The base is going to be covalently bonded to the sugar at the 1' carbon, always. And the phosphate group is going to be covalently bonded to the 5' carbon of that sugar. Okay, so I just want to make sure you understand that structure. I'm going to ask you to draw it. You'll be drawing this in lab too. Later in the semester, you'll be drawing it in lab. A little bit later. Okay? Actually, you'll be drawing it a couple times. We talk about DNA throughout the semester. So again, here it says the 1' carbon on that sugar attaches to the base, and the 5' carbon on the sugar attaches to the phosphate group. So there's another picture of that right here. Okay? So again, one difference between DNA and RNA is the sugar. Second difference are the bases. So in DNA, we find the bases adenine, thymine, cytosine, and guanine. Those are the 4 bases found in DNA. Now RNA is very similar. There is guanine, adenine, and cytosine, but RNA does not have thymine. In its place, it has uracil. Okay, so just a little bit of a difference. I also want you to know the difference between pyrimidines and purines. And so adenine and guanine, I call them the double ring bases. Okay, they're going to be purines. And cytosine, thymine, and uracil are in the category of pyrimidines. So again, these only have a single ring. Okay, so you want to know that as well. Okay, so let's focus on DNA for a few minutes. Okay, so DNA is the genetic material. DNA houses genes. Okay, and this is true for all living things. So bacteria, they have DNA. It doesn't look exactly like ours. It's a circle instead of linear. Ours is linear, but they have it, and it's made of the same stuff. Fungi, protists, plants, animals, we all have DNA as our genetic material. So DNA contains genes, and those genes code for proteins. DNA, if we're talking about a eukaryotic organism, so we briefly in chapter 1 talked about the difference between prokaryotes and eukaryotes. So prokaryotes include organisms like bacteria that lack a nucleus, where eukaryotes are organisms like animals, plants, fungi, and protists. We have a nucleus. And so in eukaryotic organisms, our DNA will be found in the nucleus, where in prokaryotes, like bacteria, they don't have a nucleus, but they do have a region called the nucleoid. And in chapter 4, we'll look at that. Okay. This is a typo. We got to remove that. Make a sketch of adenine. That was meant for something else. Okay. What else about DNA? Let me back up one. DNA is double-stranded. And so here, you're probably familiar with seeing this. This is a double-stranded helical molecule. If I look at it just in 2-dimension without that, this is what it looks like. So this is just a short little stretch. So this is one side of the molecule, and this is the second strand of the molecule. Okay, I want to point out a couple of things on here on the next slide. Okay, so there's that same image. So again, I want you to look at the sugar. The sugar here is deoxyribose. So let me show you the sugars. Here's a sugar here, a sugar here, a sugar here, and a sugar here. Okay, so we have 4 nucleotides here. Let's just go over what a nucleotide is again. So this is a phosphate group, a sugar, and this is the base thymine. Here's the next monomer, phosphate group, sugar, and here's the base guanine. These two are going to be connected to each other through a covalent bond right here. These were put together during dehydration synthesis. Okay, so we call this the sugar phosphate backbone. If I go to the other side of my molecule, I'm going to find the same thing. Phosphate, covalently bonded to a sugar, covalently bonded to a phosphate, covalently bonded to a sugar. Okay, the 1' carbon on our DNA is always going to be attached to a base. And the 5' carbon, which is here, is attached to a phosphate group. Okay. Notice how we got this 5' and 3' written. That's the directionality of this strand. And so if we look at our sugar, it goes 5' carbon, 3' carbon, 5' carbon, 3' carbon. So we say that this strand here is running in the 5' to 3' direction. The other strand, it's parallel to it. However, it's anti-parallel. So what does that mean? It's running parallel to it, but it's running in a different directionality. So again, if we look at the sugars, it's running 3', that's the 3' sugar, to 5. 3 to 5. And so we say that this strand is running 3 to 5'. So this term here, anti-parallel, that's what that means. And again, we'll see that again in some later chapters. All of the bonds in the backbone, so on this side in the backbone and on this side in the backbone, they're all covalent, but look at these dots in between the bases. These are hydrogen bonds, and we've seen this before when we looked at water. So whenever you see those dotted lines, that's a weak hydrogen interaction. They add up in space and time to become relatively strong. So I want you to see this. So this is DNA again. Adenine, thymine, guanine, and cytosine. This is a rule that you have to know. Here it's called complementary base pairing. Adenine is always going to base pair with thymine and nothing else. And guanine is always going to base pair with cytosine. So we say A to T, C to G. Guanine never base pairs with adenine. Cytosine never base pairs with thymine. Cytosine never base pairs with adenine, and guanine never base pairs with thymine. So that's a rule, okay? Between guanine and cytosine, there are 3 hydrogen bonds, and between thymine and adenine, there's only 2 hydrogen bonds. Okay, let's quickly just talk about RNA. So I got kind of a lot going on in this figure. So the RNA is actually right here. This is one type of RNA called mRNA. Then there's another type of RNA right here, tRNA, and there's actually a third type called rRNA. For our purposes right now, we don't need to know the 3 names, but we will talk about those later. What I do need you to know is that RNA is just a copy of a gene, okay? So it's not this big, long molecule. I mean, it's still a macromolecule, so big comparatively. But if you compare it to DNA, it's not big, right? DNA is thousands of genes long, where RNA is usually just a gene long. So it's much shorter RNA. RNA is also single-stranded, where DNA is double-stranded. RNA is also involved in protein synthesis. So what will happen is an RNA transcript, a copy of a gene, is made in the nucleus. That RNA leaves the nucleus, goes to the cytoplasm, and this thing here, called the ribosome, is going to read the RNA, and it's going to build a protein. So all these little balls here, these are all amino acids. This is the growing polypeptide. And so these amino acids are put together according to the message. That's what mRNA stands for, messenger RNA. And this RNA was made from the gene, so from the DNA. Okay, so RNA is single-stranded. It's much shorter than DNA. The sugar here is ribose instead of deoxyribose. And lastly, RNA is going to have A, G, and C, but it doesn't have thymine. In its place, again, it has uracil. And the last thing I want to show you, this is called central dogma. And again, we're going to come back to this in later chapters, but just to introduce it to you, the process, so here, let me just give you an idea of what this picture is. This is a cell, and so this is the cytoplasm. I'm not showing you the plasma membrane, okay? You just have to assume it's there. Here's the cytoplasm. This is the nucleus in purple. This is the nuclear membrane. So it's a double membrane organelle. It's pretty neat. And then we have our double-stranded DNA here in blue, and here we have our RNA transcript in red. And so what's going to happen is a segment of the DNA, where the gene is, will be read, and a transcript, this mRNA here, this message, will be produced. That process of making RNA from DNA is called transcription. What happens next? That transcript is going to leave the nucleus out of a structure called the nuclear pore, which we'll talk about in the next chapter. Out here in the cytoplasm, the second process, which is called translation, the RNA, which is shown here now, will be translated. So what does that mean? It's going to be read by a ribosome, that's this brown globular thing, and as it's being read by the ribosome is going to help put amino acids together in the right order to form the appropriate polypeptide, which will then fold into its proper three-dimensional shape. And if it is a quaternary, if it has quaternary structure, it'll then associate with other polypeptide chains. Okay, that's it for this tail end of the chapter.